Hi everyone, so today we are going to be looking at RNA. So we're going to be doing the entire deep dive all into RNA, which is quite exciting. So essentially, right, we're going to look at two things. We're going to look at the structure of RNA, and then we're going to look at the formation of RNA. Finally, we're going to look at the location and then the types of RNAs. It's kind of going to be a little bit of a hectic episode, but nothing that you can't handle. First, let's talk about the RNA structure, right? Because it's probably the most important. RNA has the exact same structure as DNA, although there are a few differences. So let's just draw that. So this pentagonal shape here is our pentose sugar. But it isn't just any pentose sugar. It is a ribose pentose sugar. It's important that you say ribose because that is what differentiates this pentose sugar from a deoxyribose pentose sugar. I just need to clean my pen. Cool. Then we still have our phosphate sugar. Oh, well, just our phosphate basically. And then we still have our nitrogenous base. However, what changes in the nitrogenous base is that we're still going to have adenine, we're still going to have cytosine, and we're still going to have guanine. But adenine is instead going to bond to uracil instead of bonding to thymine. And just the general structure of RNA, it's a single-stranded molecule and it's a polymer made of monomers, and those monomers are known as nucleotides. So, how exactly is RNA formed, right? So, how is RNA formed? So, a ribose sugar of one nucleotide is going to form a bond with the phosphate group of another nucleotide. So, our ribose sugar is going to form a bond with the phosphate group of another nucleotide. Then, what else is going to happen? One single long strand forms, and that RNA strand is going to consist of a sugar phosphate backbone. and complementary base pairing can take place in any number and ratio. Remember that our RNA can be located in the nucleus but it can also be included in the cytoplasm, and it forms a part of ribosomes. Ultimately, we also get different kinds of RNA. So we get mRNA, which is messenger RNA. We get tRNA, which is transfer RNA. And we also get rRNA, which is ribosome RNA, mRNA. What does each one do, right? So first let's describe the structure of each of these molecules of each of these kinds of RNA that we're dealing with, right? So messenger RNA is a single strand with an unlimited number of nucleotides. Transfer RNA consists of a single strand that folds back on itself like a hairpin. And ribosomal DNA consists of a single strand. So, what are the similarities between all three types of these RNA? It's that they consist of a single strand with an unlimited number of nucleotides. But what changes in the tRNA, is that the single strand looks like a hairpin and folds back on itself like a hairpin. So let's go into mRNA and actually discuss what that is. So mRNA is formed in the nucleoplasm. Where's our nucleoplasm? This is our nucleoplasm. And it uses DNA as a template. So mRNA, remember, it's a single strand with an unlimited number of nucleotides. It is formed in the nucleoplasm and it uses DNA as a template. What mRNA is going to do is it's going to carry genetic code from the DNA. So from the DNA in the nucleus to the ribosomes in the cytoplasm. So here. So what mRNA is going to do is it's going to carry genetic code from the DNA strands in the nucleoplasm to the ribosomes in the cytoplasm. And so therefore it acts as a messenger between our nucleus and our cytoplasm. Whereas what our transfer DNA is going to do is it actually just occurs in the cytoplasm. So it is all over here. And our transfer RNA has 3 exposed bases on one of the loops. So if a loop looks like this, for example, let's say we have bases, say A, C, G, right? So it has 3 exposed bases on one of the loops. And this is known as an anticodon. So 3 exposed bases is known as an anticodon, and it plays an incredibly important role in protein synthesis. What this anticodon does is that it picks up amino acids in the cytoplasm and takes them to ribosomes. So it picks up amino acids in the cytoplasm and takes them to ribosomes, which is why it's known as a transfer molecule. Finally, we have ribosomal DNA, which essentially just forms a part of the ribosome in the cytoplasm. So essentially, our RNA function is going to be that it carries instructions from DNA in the nucleus to ribosomes of the cytoplasm to control the synthesis of proteins. and to make sure we are also forming amino acids. So ultimately, that's it for RNA.